Everybody, thank you all for coming. Today, we have John Walter giving our seminar. John is a quantitative ecologist whose research focuses on the dynamics of aquatic ecosystems and the organisms inhabiting them, particularly in the light of global change. His work covers a spectrum from advancing ecological theory and statistical methods to studies addressing specific problems in conservation and environmental resource management. Context for his work include movements of California native fish, kelp forests on the California coast, and water chemistry and algal blooms in lakes. John got his bachelor's degree at Gettysburg College and then did a PhD at the University of Virginia, and is now a senior researcher at the Center for Watershed Sciences here at UC Davis. And today he's going to talk about his work on synchronous dynamics in giant kelp forests.
Awesome. Thanks so much for that introduction. Thank you for the invitation. It's awesome to be here. I've shared this with folks before, but I moved to California about 3 weeks ago, and so it's really exciting to have the chance to make some connections with folks and just to see a different part of the coast. This is my first time in this area. Yeah, so I guess along with that, I was really happy to connect with graduate students and faculty and stuff like that while I was here. If we didn't get a chance to chat, but you see something that you think is cool, feel free to reach out to me. I'm happy to meet over Zoom or back in Davis or whatever. Awesome. So the work that I'm going to present today is part of a collaborative project that's supported by NSF and that also benefits from long-term study at the Santa Barbara Coastal LTER. And this is our team. Some of you might recognize some folks that have Bodega and UC Davis connections, including Max and Vadim, who did some graduate work with Marissa Baskett. And I'll credit some co-authors on specific studies as I talk about them, but really the work that I'm talking about today benefits from all of these folks, and I'm grateful that we were able to put together such a fun team for this work. I mentioned that I am new to the area. I'm also a quantitative ecologist who doesn't have a field program. So I suspect that many of you might have more experiences in person with kelps than I do. But just to give a brief introduction to this system, what we're talking about is canopy-forming macroalga. It's widely distributed, and then from Baja California north through the Central Coast, it's the dominant canopy-forming kelp species, transitioning, as probably many of you are familiar, to dominance by bull kelp as we go north. And like other canopy-forming kelps, giant kelp is the foundation species of a diverse and productive coastal ecosystem. And importantly for the work I'm going to share today, the fact that it forms a surface canopy means that it's amenable to satellite measurement. And this means that we have a really uniquely spatially extensive, but also spatially detailed, and both long-term and finally temporally revised data on the distribution and abundance of kelp forests. And in large part, this is due to the work of Kyle Kavanaugh and Tom Bell, who are part of our team for this work. To give a sense of direction of where I'm headed, I'm going to formally introduce this concept, synchrony, and then I'm going to begin applying it to kelp by highlighting some prior work that uses some kind of more established statistical approaches to study spatial synchrony in kelp before getting to the meat of my talk, which is going to be about two recent studies that have a little bit of conceptual and methodological newness that provides some new insights on the dynamics of kelp forests and their influences on adjacent ecosystems. So what is synchrony? Even if you've not thought much about this in a scientific context, its colloquial meaning is likely familiar to you, bringing to mind things coinciding in time or happening simultaneously. And in this research context, we can expand that out a bit further to variables that we care about, having fluctuations that are positively related through time. And that definition is still intentionally broad because ecologists have studied synchrony in a wide range of contexts and at different organizational scales, from phenology to population dynamics, community interactions, and even biogeochemistry. And so it's sometimes complicated that these different communities don't always talk to each other. They use some different language. They have different conventions for how to analyze data on these questions. But nonetheless, this idea generalizes across a wide range of contexts different systems, different taxonomic groups. Throughout this talk, I'm going to focus on a particular kind of synchrony, spatial synchrony, and this refers to when fluctuations in some focal measure are correlated through time across locations. And that's exemplified here by the tendency for time series peaks and troughs to coincide in the middle column. And that stands in contrast to the far column labeled asynchrony, where there's no such tendency for those time series peaks and troughs to be aligned with one another. And often in ecology, we've focused on those measures of things being things like the abundance or the growth rate of populations. In this talk, largely we're going to be focused on the biomass of giant kelp, or an index of the biomass of giant kelp that we can derive from satellite imagery. So one reason that we care about synchrony is relative to the case where locations fluctuate asynchronously or independently. The total across all those sites is going to have higher variance because the synchronous components of oscillations reinforce each other in the total, whereas the asynchronous ones tend to cancel out. And you can see that here by comparing the thicker black lines in both those panels that represent the sum of those thinner colored lines near the bottom of the panel. So the upshot of this is that if we want to think about how local processes scale to regional totals, then spatial synchrony is a key phenomenon to care about. And then synchrony is also believed to increase rates of extinction and to slow recovery from extirpation because simultaneous rarity of nearby populations, like you see on the left but not on the right, at simultaneous rarity inhibits rescue of local populations via dispersal. So we might also want to be concerned about spatial synchrony when we're thinking about conserving rare organisms or in recovering from disturbances. At this stage, you might be wondering, well, like, why does this occur? So a primary mechanism is the action of environmental drivers that are themselves synchronized or spatially autocorrelated over large areas. And for kelp, these can be things like ocean temperatures, nutrient concentrations, and waves. And in the literature, this phenomenon, whereby synchronized environmental drivers synchronize a biological quantity, is often referred to as the Moran effect. Synchrony can also arise from the coupling of sites together from dispersal. And we also know that synchrony can be transmitted through species interactions like predation and competition. This is not as well studied, as well understood, and I think it's typically fair to think of environmental drivers or Moran effects and dispersal as sort of ultimate causes of synchrony, ultimate mechanisms that synchrony originates from. All right, so getting into some data. So here's some actual data on kelp that comes out of a 2013 paper led by Kyle Kavanaugh that was focused on the Southern California mainland coast. So in the map on the left, these black shaded areas where the coastline looks thicker, that represents the extent of kelp cover. And the red crosses are places where there are some field-based measurements, including the Santa Barbara Coastal LTR sites up near the top there. And then on the right is a satellite-derived record of kelp abundances across the coastline. Let me see. Okay, cool. Yeah, so, and that runs generally from north at the top of the panel, south at the bottom. And the color indicates estimated kelp canopy biomass. And just visually, you can see some synchrony. There's some apparent crashes, like this area in blue. That's relatively synchronous across a handful of sites there. And then periods as well where we see periods of high biomass that tend to be shared across at least some nearby locations. So that is an example of what synchrony can look like in some real data. And then in the new work that I'm going to talk about, we're essentially using an updated version of this kind of data. So one of the things that we can do with this, and for about 20 years, a prevailing approach to studying spatial synchrony, is to measure the correlation between all those pairs of time series of kelp biomass for all those locations, and then plot the relationship between the magnitude of synchrony and the distance between locations, like you see here. And note here the difference in the x-axis scales. So we can see a common pattern where synchrony starts out relatively strong and becomes weaker as the distance between locations increases. And from analyses like this, we can make some kind of general but imprecise inferences about the causes of synchrony. So some key environmental variables that we know from other work influence kelp, like temperature and wave height, are positively autocorrelated at these regional scales. So we can kind of loosely attribute this longer range spatial synchrony to correlated environmental drivers. And then at local scales, synchrony also decays pretty quickly over scales that are similar to scales of spore dispersal and autocorrelation and recruitment. And we can infer from that, again, kind of tentatively, that dispersal is contributing to synchrony in this system. But a big reason that these inferences are kind of loose or tentative is that we've essentially collapsed synchrony into a single axis of variation according to distance. And so a large part of my work over the past several years has been to develop and apply statistical approaches that improve inference into patterns and mechanisms of synchrony that leverage other dimensions of variability in the data. And so the two examples that I'm going to talk about today are going to feature a little bit of that, but there are also some dimensions that go beyond this that I'm not going to have a chance to talk about in this presentation. Okay, so the first new study that I want to dive into is on how tail-dependent spatial synchrony in giant kelp arises from nonlinear effects of environmental drivers. And by tail-dependent, I mean that synchrony might be stronger or weaker at either end of the abundance distribution. So either when populations are both rare or when both are abundant. So in the middle, we have what is likely a familiar looking strong positive correlation between the abundances of two populations. And then on the left, when the relationship is tighter, when both are rare, we're going to call that lower tail dependence. And on the right, where the relationship is tighter, when both populations are abundant, we're going to call that upper tail dependence. So the starting point that we used for measuring tail dependence is called the partial Spearman correlation. I'm a statistical ecologist. I need to throw some math in here. It's not super important. The important thing to gather from the important point that I want to make is that this is just a scheme that allows us to partition the ordinary Spearman rank correlation into contributions from different quantiles of a joint distribution between two variables. And so what this allows us to do is measure the strength of association within an upper band of observations and then a lower band of observations. And in this example, we see the relationship is stronger in the upper bands. And then we can use the difference in the partial Spearman correlations between those two bands to get a measure of tail-dependent strength. Okay, so now I've kind of introduced this concept. You have a notion of how we might quantify it. I'm going to sketch a theory of why spatial synchrony could be tail-dependent, and then investigate how that theory applies to giant kelp. So to begin, let's just imagine that we are looking at two populations. These can be kelp populations. They're in two different places. They're influenced by an environmental driver that is similar in those two places. Its fluctuations are synchronized, and they span a similar range of values. And lastly, that the environmental driver has a threshold-like relationship with population abundance in those places. But any pair of places that we look at might be different parts of the coast. Maybe one's in Southern California and others in Central California. Any pair of places don't experience the full range of possible values of the environmental driver. So some pairs experience this blue range, mostly low values. Others experience this red range with mostly high values. And then for the blue range, the effect of that environmental driver is going to be stronger as it's experienced at those sites in the upper tail of its distribution. And in the red range, the effects are going to be stronger in the lower tail. And that yields relationships between environmental drivers and populations that look like this, exhibiting upper and lower tail dependence. And so what I've added to the equation here are some scatter plots showing the relationship between an environmental driver and some population measure. And the two different populations are indicated by the filled and open circles. That's going to translate then, when we look at the relationship, the synchrony between those two populations, into tail-dependent synchrony in the same manner as the relationship with the environmental driver. And visualized another way, that translates to synchronous booms and asynchronous crashes when there's upper tail dependence, and asynchronous booms but synchronous crashes when there's lower tail dependence. So this theory is going to allow us to make some predictions that we can test if there's a real-life system that matches enough of the prerequisites for the theory to apply. So if different groups of locations experience different ranges of the environmental driver, then we're going to see geographic patterns in the tail dependence of the driver-response relationship. There's also going to be geographic patterns in the tail dependence of spatial synchrony between those populations, and these patterns are going to correspond with one another. And we know from prior work that key elements of that theory are fulfilled when we look at giant kelp. So there's a threshold-like relationship between kelp and waves. And here we're expressing waves in terms of wave calmness, so that there's a positive relationship between wave calmness and kelp abundance. That satisfies some requirements of our formulation of the partial Spearman correlation. In practice, wave calmness is just wave height multiplied by -1. Right, so larger and more energetic waves dislodge kelp, so it would be a negative relationship on the natural scale. And then we also know that coastline segments vary in their wave exposure, right? So we have this phenomenon where we have some wave exposed sites and some calm sites. Okay, so now let's look at that first prediction from theory about tail dependence in the relationship between kelp and waves. So that's mapped across our study area that goes from San Diego to a bit past Monterey. And I've highlighted in insets a couple of stretches of coastline where kelp cover is especially consistent, which also show us that different associations, different tail dependences, positive or negative, kind of predominate in areas that have different degrees of wave exposure due to the orientation of the coastline. So now to that second prediction from theory, this map shows tail dependence in spatial synchrony averaged within 25-kilometer sliding windows up and down the coast. So the scale here is a bit depressed by the spatial averaging, pulling everything towards zero, but nonetheless, we see areas where synchrony tends to have upper tail dependence and others with lower tail dependence. And these seem, if you remember much about the previous slide, at least passingly similar to the maps for the kelp-wave relationship. And then if we do put them side by side, we see some visual correspondence between the two. And a statistical test corroborates that the correspondence between these two maps is greater than we would expect by chance if there was no relationship between them. And then adding further support for that theory, we see a significant relationship between the time-averaged wave calmness or wave height and tail dependence in the relationship between kelp and waves, such that locations with high average wave calmness are more likely to show stronger effects in the lower tail and vice versa. All right, so just to kind of wrap up this section of my talk, we've seen that giant kelp shows tail-dependent spatial synchrony, and this can be attributed to a nonlinear relationship between waves and kelp abundance. One neat thing that I think that I didn't emphasize before is this relationship, or the difference between whether booms or crashes are synchronous or asynchronous, depending on the direction of tail dependence. This suggests that areas with upper tail dependence might be more resilient because those crashes are systematically less synchronous. And so that's going to leave likely some nearby populations that are reasonably abundant. Those might be seeds for recovery following a crash. And then we think that this phenomenon by which, I guess both phenomena by which, where there are tail-dependent relationships between environmental drivers and biological or ecological response variables and tail-dependent synchrony is probably a lot more common than we understand at present. And this is because the key ingredients of our theory are fairly common. We know that spatial synchrony, to a reasonable first approximation, is ubiquitous in nature. And nonlinear relationships are between environmental drivers and ecological or biological responses are fairly common as well. We focused on this sort of threshold-like or sigmoid relationship in this talk because it matched this dynamic in kelp, and also because it's probably the simplest functional form that can give us switching between areas that are positive, have upper tail dependence and lower tail dependence. But the idea that relationships can change in strength depending on where you fit along that environmental gradient in when you have a nonlinear driver-response relationship holds to other kinds of nonlinear relationships. And one that might be really interesting to investigate, but I'm not aware of work, at least going into dimensions of synchrony, are things like thermal response curves and potential implications for climate and ocean warming. Cool. So shifting gears a bit, one of the big questions that we had in this project is what are the consequences of synchrony in this critical foundation species that might cascade throughout the Rocky Reef community and even to ecosystems that are coupled by flows of matter and energy. So if that former, those cascades throughout the community interests you, Amanda Lohman, who is a postdoc supported by this project, is wrapping up some neat work on that question right now. I'm going to talk about some of our recent published work on the latter, on how synchrony cascades across ecosystem boundaries due to resource subsidies. So resource subsidies, by which we mean flows of nutrients and organic matter across locations and across different types of ecosystems, connect those systems and shape their structure and function. And this figure that I've borrowed from a paper by Gunand et al. Shows us that cross-ecosystem spatial flows of carbon, which are shown in pink. The specific axis labels are probably too small to read, but the ones in pink are all cross-ecosystem flows, or spatial flows, excuse me. They're common, and their magnitudes can rival and even exceed the magnitudes of local processes, which are shown in green. And so our work here is asking whether resource subsidies can produce spatial synchrony in the dynamics of the system receiving subsidies. And our study focuses on coupling between kelp forests and sandy beaches, where beach wrack, like you see in the photo on the top right there, That rack from kelp detritus provides an organic matter subsidy that is going to really dominate local beach primary production and form the base of a multi-trophic beach food web. That includes things like microbes and invertebrates and small mammals, and importantly for our study, birds. Recall, if you will, that the standing stock of, kelp biomass is spatially synchronous, especially about 50 kilometers. So we know in advance that there's some degree of synchrony in the production of organic material. How does that translate, or does it even translate to the rack on the beach? So to understand that, we focused on three main questions. How synchronous are resource subsidies, that rack on the beach? Can synchrony be attributed to synchrony in the donor system? So can we kind of trace a path of causality from the reef to the beach? And then do synchronous resource subsidies propagate to higher trophic levels? Sort of indicating an ecosystem-wide effect, not one that's just limited to the base of the system. So we used, for this study, monthly monitoring data from 5 beaches that were sampled by the Santa Barbara Coastal LTR site in California, coupled again with our satellite remote sensing-based data on kelp abundance on the offshore reefs. And in this map, the green areas are kelp forests, and the 5 beaches that we studied are indicated by black points. And at those 5 beaches, we have monthly measurements of an index of the quantity of rack on the beach. So to better understand, I want to take a slight detour into this notion of timescale specific synchrony. So you might have noticed that we're studying monthly time series in this work. And so they're going to contain some patterns that just reflect seasonal cycles. This is also a long-term study. And so they're also going to contain signatures of longer-term oceanographic oscillations like El Nino or PDO that make entire years or even stretches of years favorable or less favorable for kelp, and a whole host of other things too. But how we're going to deal with that statistically so that those different timescales of variation don't become conflated and hinder inferences. So as a toy example of this phenomenon, let's just focus first on panels A and B. These show pairs of long and short timescale oscillations respectively. If you come from a more strongly physics-based discipline, you might be more comfortable thinking of these as low and high frequency. So in panels C and D, we've combined those time series on the left in particular ways so that they have perfect synchrony, perfect positive correlations on some time scales, but are perfectly anti-synchronous or perfectly negatively correlated on others. And what this illustrates is simply that systems can differ in their dynamics depending on the time scale of variation that you are focused on. And a second point here is that if we were to measure the ordinary correlation between each pair of time series in those 2 panels, it would be 0, or to within a rounding error of 0. And what that illustrates is that sort of our conventional tools for evaluating relationships and studying synchrony, which we've applied for a long time, are really kind of ill-suited to this phenomenon of timescale specific synchrony. So a somewhat contrived example, but hopefully a decent illustration. So many of the tools that we have for dealing with this challenge of timescale specific synchrony are based on wavelet analysis, including this technique, which we call a wavelet mean field. So this is totally unrelated to kelp. It's actually for dissolved organic carbon concentrations in a bunch of lakes. I wanted to show it, though, to kind of introduce this method, illustrate how to interpret it without giving away the kelp story. So what the wavelet mean field does is it combines the information about variation in each time series that are encoded in individual wavelet transforms in such a way that synchronous oscillations, the ones that have similar magnitudes and their peaks and troughs are aligned, are going to take large values. That's indicated by the warmer colors. And then this is going to be both timescale specific and localized in time. So we can see specific timescales or frequencies of variability, and also whether this is consistent through time or potentially changing. So this gives us heat maps that look like this. We have time along the x-axis, time scale along the y, running from short time scales or high frequencies at the bottom of the page to longer multi-year oscillations toward the top. And then the black contouring that you see in this panel is just going to be an indication of statistical significance. Okay, so now back to kelp. So our first question was, is RAC synchronous? And we see here that RAC abundance on the beach does exhibit spatial synchrony, and with different dynamics across a few bands of timescales. So from 2 months to 8 months, so the shortest fluctuations that we can resolve in the data, to about 8 months long, we see synchrony that is kind of episodic. It's not consistent across the time series, but there is significant synchrony that is associated with certain potentially events. At 8 to 16 months, which kind of encompasses our annual cycles, we see a band of quite consistent synchrony running throughout the time series with a little bit of variability and disruption, but overall quite consistent. And then we also see pretty strong synchrony at some of the longest time scales that we can resolve in this data, about 3 to 5 year cycles. So now we wanted to answer question 2 about attributing RAC synchrony to causes. And so we extended on this wavelet mean field method by building a multivariate model of RAC synchrony as a function of synchrony in kelp supply. So there's our sort of offshore production variable, wave action, and beach condition. So wave action is kind of controlling, well, what is the wave environment doing? Is it dislodging kelp, dislodging fronds, entire plants? And then beach condition is telling us, which we expressed as, so the width of the dry beach on this system, basically how receptive is the beach to receiving kelp RAC? And if we don't have any dry beach, we can't retain kelp on the beach. If the environment is too reflective, then it's going to get washed out to shore. So panel B shows the predictions from that model. And so you can see that there's a pretty strong visual correspondence, at least for the sort of annual timescale and the multi-year timescale bands. It doesn't do such a great job at our shortest timescales. And yeah, feel free to ask me about why I think that is. And then panel C is breaking down the contributions of those 3 main terms in the model, showing only statistically significant ones, as well as interaction effects, which in this case are all kind of either negligible or they're antagonistic. So in this case, there are some relationships between our predictor variables, and so actual synchrony is less than would be expected if the driver variables all acted independently. That's kind of what those interaction terms indicate. So our third question asked whether RAC synchrony propagates across trophic levels to synchronize the observed abundances of shorebirds that forage in the RAC and feed on RAC-associated invertebrates. So following the same kind of format that we just saw, and starting with panel A on the top, we see synchrony and shorebird abundances across beaches in the wavelet mean field, similar to what we saw before. It's more episodic at our short, sub-annual timescales, more consistent, though not entirely so, across our timescales representing annual cycles that arise from seasonal variation. And then we also have some synchrony at long timescales as well. Moving to panel B, we have our predictions. Again, it does a pretty good job of recovering the patterns of synchrony that we see in empirical shorebird abundances, especially at those annual timescales. And then panel C on the right, again, is showing the contributions of terms in the model. Here, the terms are a little bit different. We included air temperature as a way to account for the fact that these birds are migratory. Air temperature is probably not ultimately the cue of migratory behavior in these birds. There's some literature to suggest that air temperature can influence sort of local variations in migratory timing. But here what it's really doing is it's sucking up an annual cycle. And what this does is actually improve some of the tests of assumptions that underpin this modeling. And so we included it even though it's in some ways sort of a dummy variable. But it makes our models work. So that's a good thing. And then again, I'll just point out that the annual band was just the only set of timescales that the model was statistically significant, even though it does explain a decent amount of variability. And then last point here is that we often have thought about spatial synchrony in sort of demographic processes, so changes in abundance that arise from births and deaths and dispersal. Here, we're really talking about a behavioral response, where these birds are choosing to forage in areas with more rack because of greater prey availability, as opposed to that demographic one. All right, so kind of pulling this study together, we've kind of traced some causality that looks something like this. So we have kelp biomass being produced offshore, waves come in and dislodge kelp, and currents transport it to the beach. The beach condition, especially its width, mediates the retention of rack, and then shorebirds forage where rack is abundant due to prey availability. On its own, none of these things is particularly new, which in some ways is a good thing because it shows us that we're not completely off base. But what is new is the demonstration that these mechanisms act synchronously across sites, and that results in this transmission of spatial synchrony from kelp forests to sandy beaches, and then that transmission of synchronous shorebird predators supports that this is something that has ecosystem-wide consequences. So looking beyond the study, I think there's some pretty neat implications. So it's likely that this phenomenon whereby resource subsidies drive spatial synchrony is at least somewhat common. I mentioned before that spatial synchrony is quite common. And we also know that cross-ecosystem flows of carbon and nutrients are also common, and they can be big relative to local production. That's when we'd be most likely to at least be able to detect this. So this is potentially an important driver of the spatiotemporal dynamics of recipient ecosystems, so those including sandy beaches that really depend on subsidies because local in situ primary production is pretty low. And finally, I'd like to highlight that subsidy-dependent ecosystems are vulnerable to changing patterns of connectivity and donor ecosystem production, and something that could likely be shifting and appears to be as oceans warm and disturbances like marine heat waves become more common and widespread. So wrapping things up, I'd just like to reflect on the fact that kelp forests are really amazing systems for developing and testing ideas about synchrony, and that the possibilities for applying these ideas and methods seem just about endless for kelp and for many other kinds of study systems. So all that said, one area where I think that we don't yet know enough, and that I see as a potential future direction for my work, is how to apply synchrony to the practice of things like ecosystem restoration and management of exploited species. Early in my talk, I showed two contexts where people who study spatial synchrony have often argued that they should be really important for conservation and management concerns. But my experience in working with practitioners is that these ideas aren't necessarily translating to those types of contexts. And so what can we do to kind of bridge what has largely been a conceptual and somewhat theoretical interest with on-the-ground work? So thanks very much for listening. I'd be happy to take any questions. I think we've got a bit of time. Thanks very much. Are you just waiting for the cool cube, or have I stunned you into silence?
I can ask.
That means either my talk was brilliant or was incomprehensible, and there's no in between.
Does it work? Oh, I have a question. The one that you asked, but why at the small time scale? And also, why do you think there was such a
Yeah, okay, so I'll tackle the first question first, which was about, why does our model do poorly at short time scales? And I'll scoot back to that slide so we can see that. Yeah, so you can see, in the model predictions, and then in the on the right, indicating the contributions of those terms to synchrony. It's really not excellent. So I think there might be a few things going on here. So one possibility is that there's some variable that is responsible for dynamics on short time scales that we did not include, didn't think to include. Another possibility is that even though we have monthly data, there are things that are happening on finer timescales than that, but are having somewhat lasting effects. So these could be shorter, more episodic storm events and things like that that are particularly severe, that then impact kelp directly, impact RAC directly, or sort of kickstart some kind of recovery process. That's thinking a little bit more on the sort of supply side of the equation. On the beach side of the equation, it's one thing that we know. And I really mean the people who study that system in the field, like Kyle Emery and Jenny Dugan, know, is that the particularly juicy parts of the kelp plant, so particularly the blades, get consumed really quickly when they're deposited on the beach. And so if the timing of sort of deposition versus the timing of when the sampling took place is a little bit off. That could be a source of noise in the data that we just can't really account for. There was a second part of your question. Yeah. Yeah, so when we're getting into like 3 to 5 year timescales, that is getting into like the timescales where ENSO is operating. So that could be one reason. Another possibility, another thing that could be occurring is that it sort of becomes like easier to synchronize those sort of longer, slower fluctuations. And you can kind of imagine that like if you have a really like long wave or like a really low frequency oscillation, and you just like nudge it a little bit off, it's still pretty close to being positively related to the one previously. But if you have a really high frequency oscillation or short time scale oscillation, and you nudge that just a little bit from one time to the next, then you're going to be kind of off. So there is a phenomenon where it's easier to detect long time scale synchrony because of that. That's one of the reasons that we do statistical tests, is because those bake that into the null hypothesis. But sometimes when we're just visualizing the data, and this is primarily a visualization tool, the wavelet mean field, we are more likely to like see synchrony and measure it as being strong, but it might be sort of weak in a more mechanistic sense, if that follows. Okay, cool. Yeah, thanks for the question. Yeah, that's a little long to chuck the cube. I was hoping for an opportunity, but my arm's not that good.
I had a question. Since I noticed that these are like really long time scales, does that like development work? And just kind of ask that, like, is there anything that would want to know right now that we don't have?
Yeah, so that's a good question. So this work is largely dependent on having long time series, and in particular the statistical methods that, especially in this second study, are quite dependent on having pretty long data, and so that does constrain some of the questions that you can ask, and constrains what systems that you can ask them in. We're really fortunate to have a fantastic exemplar system in Giant Kelp, and if it wasn't clear from the way that I talked about this work, I came to this not as an expert in Kelp, but as an expert in some of these statistical tools. And so one question that I think is particularly interesting across many different systems that is difficult to assess, in part because of lack of data, is how is synchrony changing over time? We can see some changes in figures like this, but especially if we were starting to zoom out to long time scales, like we might think of as being associated with long-term climate variability and climate oscillations, it can be really tough to understand and try and attribute mechanisms of those changes. And so that's something that is interesting.
I had two questions. One was simple. It was about dispersal and synchrony. Is it as simple as when more dispersal, you get more help, and when less dispersal, less help, or that's more complicated, but I figured it out. And then the second was the synchrony. Sorry, I'm not talking like you. What can you talk more about wave stress?
Yeah, so I'm gonna preface this by saying that I'm not a kelp expert. So like, take anything that I say specific to the kelp system with like just knowing that it's like filtered through my conversations with people who actually are. So yeah, the first question about dispersal. Yeah, so it basically, dispersal is sort of a means of like coupling the dynamics of different populations. And so if you have, in general, if you have, more dispersal between two populations, their dynamics are going to influence one another and sort of tend to like bring them both into having similar or synchronous dynamics. So it's not quite just like, is there more dispersal or less? It's a little bit like more complex than that. But I think a good way to think about it is like, if you have those, if you have dispersal between those places, then their dynamics are going to be coupled, at least partly. Yeah, and so the second part was about thermal stress. Yeah, so that's something that we didn't actually really look at in either of these studies, but in some of the other work from this project has proven to be quite important. And one of the interesting things that as kind of pertains specifically to synchrony is that the relative importance of thermal stress versus waves in creating synchrony seems to differ depending on where you are on the coast. So more in like the central coast, and I would imagine more in the north coast as well, although that wasn't part of our study area. The central coast is driven more strongly by waves. There's like a stronger seasonal pattern of strong storm-driven waves in the winter, kind of knocking down kelp, creating some local extirpations, but then like it recovers, it regrows the next year. In like Southern California, so about like the Santa Barbara Channel, and the temperature dynamics are kind of the more dominant player in terms of like which environmental driver is acting to shape the degree and the dynamics of synchrony. Does that help to get at your question? Cool.
Thanks for asking that question, because that is related to my two somewhat interrelated questions. I guess one was so my understanding is that one of the cool things about this is the spatial synchrony. But if you have areas where there's different seasonal dynamics going on with kelp, how was that accounted for in the model? Or maybe I missed that. And then two, with the data set that you have, do you have some sites that have been restored and haven't been restored? And do they follow expectations of the non-restored sites, or like the kelp beds that have been there? Is that something you can look at with this type of data set, this type of analysis?
Yeah. Okay, so the first part of the question is about like accounting for different areas. So yeah, so there are different ways that we can like address that in different ways. this study was all Southern California, and so that was mostly, so that's more of the less seasonal dieback as part of that dynamic. The first study that I talked about, because we were basically using a moving spatial window, we kind of minimized that that concern because there is going to be some variation with coastline orientation and how severe the waves are on a local scale as well. But we sort of minimize the broader geographic patterns in that by using this sliding window. In other studies, we have looked at sort of the central coast and the southern coast separately because we know that the dynamics are different in that regard. And yeah, so there's a lot of different ways that we can kind of slice and dice the data to kind of be intelligent about controlling for that. And sorry, your second question was. Yeah, thanks for that question. I think that's a really interesting question. That's not something that I've looked at and not something that we have really turned the lens of synchrony towards at this stage. But I know Kyle Cavanaugh and Tom Bell are quite interested in kelp restoration and how that's the dynamics of that, how that's progressing, and how it depends on sort of environmental contexts. So yeah, it's not something that I've been a part of, but I think it's a really cool question and kind of starts to play into some of the potential future directions that I'd like to get into with this work. So yeah, thanks for that.
Okay, there's a question on Zoom. We have time for one more question. So someone asked, what was the reason for focusing on this specific species versus a specific division or order of kelp?
Yeah, so that's a good question. So because we were using this satellite-derived data set, we were limited to canopy-forming kelps. We can't really. I mean, it'd be awesome if we could see the understory from satellites. That would open up some really other really cool questions. But unfortunately, we can't do that. And the focus on giant kelp specifically, it was really related to what the data availability was at the time we started this project. So in addition to this work, Tom Bell has continued and is now creating a global canopy-forming kelp data set from satellite imagery, basically, with some adaptation to particularities of different regions of the world, adapting this methodology to that. And so there's now potential to expand this type of work up, through Northern California, Washington, Oregon, and up into Alaska as well. And yeah, I'm not sure that we have specific plans for that at this stage, but we definitely have now the data to begin asking questions on a more global and cross taxonomic scale, and that might also open up some interesting questions, especially if we can couple that with some field data collection and the right expertise of some more genetic and evolutionary types of questions as well than we've asked so far.
Thank you so much. That was great.
Yeah, thank you guys. This is awesome.
And if you're here, you can go to Wind Down Wednesday. Yay! Once we set it up.